Hello and welcome to this parallel ESID session. This afternoon we're going to be talking about actin cytoskeletal related disorders. My name is Siobhan Burns. I am Professor for Translational Immunology at University College London, and I'm delighted to be asked to chair this session. We have three great speakers coming up, each giving us different aspects of this very interesting area. Firstly, we're going to listen to Fabio genetics and research. So Loic has worked on Wiskott Aldrich syndrome. Afternoon in his lecture, which is entitled Unraveling, after the third talk, as this is a pre-recorded session, but I hope that you really enjoy the talks. Thank you.
Hello, my name is Fabio Candotti, and I was asked to open these sessions by providing an overview of actin cytoskeleton related immune disorders. I have nothing to disclose. As a way of introduction, you know that the cell cytosol contains a system of fibers that allow the cells to maintain its shape and to change it as needed during the life cycle. Collectively, this system of fibers make up what is known as the cytoskeleton, which is composed by three main classes of filaments: the actin filaments, the microtubules, and the intermediate filaments. We will be talking mostly about the actin filaments and the actin cytoskeleton, which has become more and more relevant to the field of primary immune deficiencies. As you know, actin fibers are produced by polymerizations of globular actin monomers, which leads to the productions of double spiral fibers that are called mother filaments. Particularly important for cell motility and endocytosis, additional actin fibers can then be produced, starting off the mother filaments, but this requires nucleation provided by the ARP2/3 complex. The ARP2 complex, in turn, is to be activated by one of the Wiskott Aldrich syndrome protein superfamily nucleation promoting factors, among which WASP and WAVE. The activation of the RP2/3 complex then leads to the production of these daughter filaments. It's important to know that the polymerization of actin is inhibited by the associations of capping protein to the barbed end of the actin filaments. For cytoskeletal remodeling to happen, the polymerized actin needs to be depolymerized, and this occurs through the actions of disassembly cofactor, among which cofilin, which mediates the debranching and the disassembly of polymerized actin into monomers unit of actin that can be recycled. So as you can see, a normally functioning cytoskeleton needs to undergo a series of coordinated phases, all of which are dependent on a series of proteins and factors, which of course can be targets of genetic mutations that affect their functions and that can result in disease. In fact, we know of many diseases that affect the ability of actin to polymerize, and many of these diseases are present with features or clinical features of immune deficiencies. For example, mutations in gain-of-function mutations in the beta-actin gene cause the productions of actin monomers that are unable to bind properly to profilin, and therefore lead to a reduction of actin polymerization. And MLK, the mutations in MLK, the transcription factor MLK, result in a reduction of actin expression. Both these conditions lead to immunodeficiencies that present with recurrent infections, thrombocytopenia, and defective actin polymerization and production. Another defect that affect the production, the normal production of actin, has to do with the capping proteins that I mentioned earlier. What I did not mention is that there exists a group of proteins, the Carmil proteins, that are able to bind the capping proteins and thereby competing for binding to the actin filament, therefore increasing actin polymerization. The corollary of this is that mutations affecting the function of Carmil2 will reduce the ability of actin filaments to be produced. And this is what was demonstrated by two groups initially, and by Jean-Laurent Casanova and by Fabian Oak. They showed that Carmil deficiencies are associated with immunodeficiencies that presents with recurrent infections, atopy, EBV-induced muscle tumors, and functional T lymphocyte defects. I'm showing you here a picture from Fabian's paper that show how the amount of actin in activated T cells from one of these patients is much lower than the one observed in the healthy control T cells. A functioning cytoskeleton also relies on a normal amount of disassembly and recycling. As I said, the disassembly recycling is based on the action of proteins like cofilin, which can be activated by protein containing repeat of the WD40 domain. These proteins are such as the WDR1 protein and the choronin 1A protein. These proteins then activate cofilin, and on the opposite side, if these proteins are mutated and their function is decreased, the disassembly function of cofilin will be impaired, and actin disassembly will be affected. And this is what was seen in patients carrying or found to be carrying mutations in the WDR1 gene, both by Steve Holland and by the group of Kahn Watson. As you can see in this picture, where actin is colored in red and the nucleic acids are colored in blue, the amount of actin in the cells from the healthy donors is clearly reduced compared to the cells from the patients, in this case neutrophils. The same thing was observed by Kahn in his patients, in which F-actin accumulation in T cells, in this case, was much augmented in the patients compared to controls. Now, in addition to these defects that affect actin polymerization, there are a series of defects, important defects, that affect the branching function of the actin cytoskeleton. And these affect a series of nucleation promoting factors, among which the Wiskott-Aldrich syndrome protein. This is a well-known, the Wiskott-Aldrich syndrome protein is a protein that is mutated in a well-known disease, the Wiskott-Aldrich syndrome, that presents with thrombocytopenia, eczema, infections, malignancy, and susceptibility to autoimmunity. And it's caused by the fact that a missing or dysfunctional WASP is not able to properly activate the ARP23 complex, thereby making the productions of actin-containing structures defective, like shown in this picture, where the microvilli of a Wiskott-Aldrich syndrome T cells are compared to the microvilli of a control cells. As you can see, the ability of the Wiskott T cells to make microvilli is greatly defective. Other diseases, other genetic deficiencies that affect the nucleation promoting function of the actin cytoskeleton are WIP deficiencies, or deficiency of the Wiskott-Aldrich syndrome protein interacting protein. WIP stabilizes WASP, so in this absence, the expression of WASP is diminished, and its function is likewise diminished. Deficiency of PST-PIP1, an activator of WASP, results in a severe autoinflammatory syndrome, whereas deficiencies of HEM1, an activator of another nucleation promoting factor, the factor WAVE2, results in a immunodeficiency characterized by recurrent infections and migrations defect noticed in NK cells that also have cytotoxicity defects. Last, for these slides, I'll tell you that mutations exist, genetic diseases exist that affect directly the function of the expression and function of the RP2/3 complex. This is the topic of the third presentation in this session, so I will let Stefano Volpi tell you about that story. Now, these defects that I've shown you so far affect the cytoplasmic cytoskeleton, but you should know that in addition to the cytoplasmic cytoskeleton, actin is involved also in the formation of what is called the cortical cytoskeleton, or the cortical actin network. This is a series of structures composed by linear actin filaments that run parallel to the cell membrane, and they are fixed to the cell membrane through the cross-linking functions of the ERM proteins, asbrin, radixin, and myosin. Obviously, mutation that affects either of these proteins can disrupt this interaction and affect the stability of the cortical cytoskeleton and the membrane. And this is what happens in a disease that is caused by mutations in the myosin gene, MSN, and that results in the X-linked MSN-associated immunodeficiency that presents with recurrent infections, cytopenia, and affected both neutrophils and monocytes, as well as platelets and lymphocytes, and with additions of adhesion and migration cellular defects. These figures only gives me the opportunity to remind you that the cortical cytoskeleton is heavily involved in the stability and function of integrins, while the cortical cytoskeleton is linked to the membrane. Integrins are kept in a inhibited form, and this therefore gives gives us a link between the cortical cytoskeleton and diseases that affect the expression and function of integrins, like the leukocyte adhesion deficiency type 1. To keep talking about the membrane location and its relevance to the effects of the actin cytoskeleton, it is important to notice that a series of very important players that regulate the actin cytoskeleton reside at the level of the membrane, and these players are the small G protein family, RAW, CDC42, and RAC. These small G proteins regulate the generations of actin-containing structures like stress fibers, filopodia, and lamellipodia through the activations of specific nucleation promoting factors such as in a VASP, WASP, and WAVE. So as you predict by now, we can expect mutations to affect these proteins and therefore affecting actin remodeling and the cytoskeletal function. In fact, mutations affecting CDC42 can cause a autoinflammatory disease called the neonatal onset cytopenia with autoinflammation, rash, and hemophagocytosis. And other mutations, they are affecting genes that are in the same pathway as CDC42, are also affecting the actin-containing structures that are regulated by CDC42. This is the case of DOC8, that is a CDC2 specific guanine nucleotide exchange factor that activates CDC42, and it is in turn activated, according to some evidence, by the STK4 molecule. Therefore, STK4 deficiency and DOC8 deficiency are expected to affect the same pathway that is affected by mutations of CDC42. Similarly, genetic mutations have been found to affect the expression of RAC2. And these mutations can be loss of function and underlying immunodeficiency that presents with recurrent severe abscesses, defective wound healing, and defective neutrophil adhesion and migration. Or these mutations can be gain of function and present with a phenotype that is that of a severe combined immunodeficiency. DOC2 is listed here because it is a RAC2 specific guanine nucleotide exchange factor and specifically activate RAC2. So in the absence of DOC2, we are expecting this pathway to be affected. In fact, as you know, mutations in DOC2 and DOC8 have been described by the groups of Gigi Cotarangelo and Helen Su, and they are very well-known disorders now that presents with recurrent infections, lymphopenia, and defective lymphocyte migration and activation. So in this overview, I presented to you very quickly a series of 25 different disorders that affect different pathways of the actin cytoskeleton formation and remodeling. And if you look at them on this slide, you would certainly say that there is a lot of heterogeneity among these diseases. But I think we can start to see some common motifs here, and certainly cytopenias that affect the myeloid compartment, neutrophils, monocytes, and platelets is a common thread of many of these diseases. And the reduction in number of immune cells is also applies to the lymphocyte compartment, with many of these disorders presented with lymphopenia or in general with leukopenia. Not only there is a reduction in number, but there is very often a dysfunction of lymphocytes, be that a reduction in cell proliferation, defective cell proliferation, defective cytokine production, or a defective formation of the immunological synapse that is mainly seen at the level of NK cells. Not unexpectedly, we also find often migration defect, adhesion, and chemotaxis defect that reflect the problems of cytokine, of actin polymerizations and remodeling in these diseases. Now, Also, not unexpectedly, many of these diseases, the majority of these diseases, present with recurrent infections. And of course, if they didn't, perhaps we would not be talking about them today. But what is also becoming apparent is that many of these diseases have features of autoimmunity and or autoinflammation, which certainly need to be further investigated, but it seems to be a common feature of this group of diseases compared to other disorders of congenital disorders of immunity. Then I wanted to conclude by showing you these slides that list a 40 or so genes that are responsible for the correct functioning of the actin cytoskeleton in B cells. And as you can see, I've highlighted just the 4 genes that are now known to be associated with immunodeficiency involved in the cytoskeleton. And this is just to say that there is a lot of work still to be done in this field, some of which I'm sure will be presented by the speakers that will come after me. So I'll stop here. I'll thank you all for your attention, and I'll be prepared to answer any questions if there are any. Thank you.
Good afternoon. I'd like to thank the organizers for giving me the opportunity to present our work in the context of this very exciting session on actin-related deficiencies. And I will focus on T cells and in particular showing you how we are moving from single disease studies to more systematic approaches. So what I find one of the most fascinating aspects of immune cell biology is the way the actin cytoskeleton is shaping very diverse morphologies in different immune cells. Diverse morphologies, but also diverse actin-rich protrusion that sustain very specialized functions. So here examples of a macrophage emitting podosomes, which are very important for matrix remodeling. Here, dendritic cells assembling very long protrusions to optimize their search for antigens. And here on the right, a cytotoxic T lymphocyte assembling an actin-rich immunological synapse at the contact with the target cell. And in addition to control the shape of individual cells, the actin cytoskeleton is also very important to induce shape remodeling during the trafficking of leukocytes. So for example, here, the example of a T cell crossing the endothelial barrier, migrating through tissue in response to a chemical gradient, and stopping migration when it encounters an antigen-presenting cell and assembles an immunological synapse. So all those steps are tightly regulated by actin cytoskeleton remodeling. It is therefore not surprising that a number of the genes associated with inborn errors of immunity are actually regulating actin cytoskeleton remodeling. Today we know about 20 such genes, which are listed in the boxes here below, which, when mutated, are responsible for some of the inborn errors of immunity. And if we look a little bit beyond the genes that directly control actin remodeling and consider genes which are involved in cell motility and or cell adhesions on the basis of gene ontology term, we may come up with nearly 1/4 of the genes associated with inborn errors of immunity related to basic cellular functions directly dependent on the actin cytoskeleton. So one important actin-related structure that we and others found to be often altered in inborn errors of immunity is the immunological synapse. So here again, the one formed between a cytotoxic T cell and a target cell. So this immunological synapse is a site of a very structured organization of multiple receptors, including the T-cell receptor, addition molecules such as LFA1 and larger glycosylated molecules. And all those receptors are actually anchored in a way or the other in the underlying actin cytoskeleton, which itself is organized in different areas at this immunological synapse. Outer area made of mostly branch actin filaments, more internal layer made of longer filaments, and there are also at the immunological synapse actin foci here associated, for example, with the TCR. So when we zoom in here using structural illumination microscopy of a synapse between a T cell and a flat surface coated with ICAM1 and TCD3, we can clearly distinguish different areas of actin reticulation with very dense actin, areas made of branch actin networks, and areas of reduced reticulation made of longer filaments that are often cross-linked. So the control at the molecular level of this complex architecture of the cytoskeleton is starting from surface receptors. There is a cascade of molecular events involving GTPase regulators, GTPases, which then in turn activate actin remodelers. And those actin remodelers, therefore, control different actin remodeling activities such as depolymerization, polymerization, cross-linking. And a good part of the knowledge we have today, at least for the immune system, of the role of multiple regulators of the actin cytoskeleton comes from our identification of genes or defective genes in some of the inborn errors of immunity. And those are highlighted here in purple. So they encompass upstream regulators of raw GTPases, such as some of the DOC GEFs. Mutations have been described also in raw GTPases themselves, as well as in multiple actin regulators. For example, WDR1, which drives the severing of actin filament, the well-known Wiskott-Aldrich syndrome protein, its interacting protein WIP, more recently described HIM1 molecule, part of the WAVE complex. And all those molecules here are actually converging in activating the R2-3 complex, which drive actin polymerization via branching activity. So our contribution to the field has mostly started by the study of T lymphocytes from Wiskott-Aldrich syndrome patients. And we've been characterizing initially defect in the organization of the immunological synapse, a defect in stability of the synapse that was associated with erratic signaling. And more recently, through multiple collaborations, we've been gaining access to deficiencies in additional actin regulators, some of which being much related to the Wiskott-Aldrich syndrome protein, including WIP, HIM1, ARPC1B. And we've been seeing that the synapse defects appear to be more pronounced in those three pathologies as compared to the Wiskott-Aldrich syndrome. We also studied recently patients with deficiency in WDR1, and here, in contrast, we found that the synapse from the T cells of those patients appear to be larger and richer in terms of actin cytoskeleton content. In parallel to studying the synapse, we've been also studying multiple related functions, including the ability of the cells to migrate, to adhere via LFA1, to degranulate lytic granules, to kill, and to proliferate. And it seems that we've got quite a diversified picture in terms of the defect stemming from those related deficiencies. So the lesson from it would be that probably those actin regulators play non-redundant function, but those independent studies would also call for more systematic approaches in order to be able to really make sense of the specific defects in those different pathologies. So I will not now spend the rest of my presentation by first zooming in the Wiskott-Aldrich syndrome immunological synapse and show you how the use of high-resolution cell imaging approaches has helped us to understand the cause and consequences of the T-cell synapse instability in the Wiskott-Aldrich syndrome. And I will then kind of zoom out this time and show you how we are now developing high-content cell imaging as a pipeline to compare different inborn errors of immunity, at least in terms of the characteristics of the related T cells, and how we believe that this can be a more systematic way to compare those deficiencies. So the way we perform our study is mostly starting from blood or PBMC from patients. We purify individual cell subsets, for example, cytotoxic T cells, expand them with feeder, and here, as an example, expansion of T cells from 3 Wiskott-Aldrich syndrome patients with no residual WASP expression. The phenotype of those cells was found to be normal in terms of expression, for example, of cytotoxic molecules such as perforin and granzyme B. But the lytic activity of those Wiskott-Aldrich syndrome CD8 T cells was defective. Here assessed in a flow cytometry-based assay, where we measure the number of residual target cells. So you see that healthy donor managed to eliminate target cells in an anti-CD3 dependent manner, and WASP patient T cell failed to do so. This killing defect was not associated with a problem of degranulation, as seen here with normal LAMP1 exposure at the surface of the cell. But we then looked at the ability of those patient T cells to migrate, find target cells, and kill those target cells using video microscopy. So what you will see here is T cells in green, and the event of death in a target cells appearing as a red dot through the use of propidium iodide. So what we could observe is that the patient T cells here on the right were actually able to migrate, were also able to encounter target cells. But like here, after 2 hours, you may appreciate that they have killed much less target cells. So we, in our quantification, could actually see that, yes, they were much slower at killing target cells. And what was very clear is that also those T cells from the WASP patient could encounter target cells. It took more time upon encounter to kill the target cells. So really, given a sense that defect is mostly a defect in the delayed killing activity. So zooming in the immunological synapse of those T cells, we could see that in the Wiskott-Aldrich syndrome T cells, they had poorer actin cytoskeleton, but also what was very striking is that they failed to enrich high affinity LFA1, this very important integrin, at the contact with the target cell. So this was also confirmed by flow cytometry analysis using a conformation-specific antibody. So we then really looked in details now at the organization of the actin cytoskeleton using turf microscopy and staining actin with sear actin. And as you see here on the right, the T cells from the patients, firstly, were failing to form those radial synapses that you see typically in normal cells. And they had a very poorly reticulated actin cytoskeleton meshwork at the synaptic plane. They also failed in most of the cases to assemble actin foci that you see clearly on the normal cells. Quantification confirmed over multiple cells that those patient cells failed to assemble a circular synapse, and here on the right that they had a defect in the density of the actin cytoskeleton meshwork through a radial profile analysis. So then we zoomed in again using this time structural elimination microscopy, and with this approach we could for the first time in normal cells look at the fine topology of LFA1 in relationship with the actin cytoskeleton. So what you see here, those red dots correspond to actually the organization of high affinity LFA1, which we found to be organized into very dense and compact LFA1 nanoclusters, which were embedded actually in the finely reticulated actin cytoskeleton meshwork. We also, through this super resolution imaging, can access to the precise localization of lytic granules. And you see here that most of the lytic granules we detected in the synaptic plane were actually located in a kind of the inner leaflet of the LFA1 nanocluster belt that assemble around the immunological synapse. So what's happening now in the T cells from the Wiskott-Aldrich syndrome patients? So as I showed you before, we found also, again, evidence for a much less reticulated actin cytoskeleton meshwork, essentially towards the center of this immunological synapse. And this led to a type of uncoupling of the LFA1 nanoclusters from the actin cytoskeleton. You see here that many of the LFA1 nanoclusters were not associated anymore with actin filaments. And it resulted also that the lytic granules appeared to not be anymore confined by this nice belt of LFA1 nanoclusters. When zooming in again, and this time using DStorm, which is an approach that allows us to look at LFA1 at a single molecule resolution, we could observe very dense and compact nanoclusters, in the case here on the left of a normal T-cell synapse. Those clusters also, although present, were much more diffuse in their distribution and less dense in the Wiskott-Aldrich syndrome patient T cells. Here, quantification showing you that the patient cells form less clusters per surface area, and that those clusters were less rich in terms of molecular density. So the conclusion from this study using different super resolution microscopy approaches showed us that in the WASP deficient CD8 T cells, the delayed killing activity was most probably due to the inability to stabilize this immunological synapse, due probably to the destructured actin cytoskeleton meshwork at the immunological synapse, not allowing the confinement of LFA1 in nicely structured nanoclusters, and not allowing the proper polarization of the lytic granules. So I will now move for the final part of my presentation to our current effort in developing a pipeline based on high content cell imaging in a way to systematically compare and discriminate defects in cells from various patients with immune errors of immunity. So we first implemented this high content cell imaging pipeline using cell line as a model, the NK92 natural killer cell model. And our pipeline is based on the stimulation and also the staining of cells directly in 384 well plates. And then we automatically image the different wells of such plates with automated confocal microscope. And this is generating thousands of pictures, typically in one experiment. So the quality of the images, so here zooming on single cell, is very good, and this is really like confocal, usual confocal microscopy. And you see here very clearly that the NK92 cells, when stimulated with ICAM1 and stimulatory antibodies, moves from a roundish morphology to a flattened morphology, emitting nice actin structure, polarizing individual lytic granules here in red. So with this approach, we can also automatically quantify characteristic features from the cells And here you see in this radar plot profile that one of the features, as expected, to be highly upregulated upon activation of the NK92 cell is the F-actin content, actin intensity, that we could measure here across thousands of cells in an automatic way. So to further validate this approach and to look at its ability to detect alteration in the actin cytoskeleton, we treated the NK92 cells with drugs known to affect cytoskeleton, cytochalasin D and latrinculin B, which were used at 3 different concentrations. And very clearly, you can see that the F-actin intensity in the synapse of those NK92 cells is affected here in terms of F-actin content in a dose-dependent manner, therefore validating this type of approach. So we then started recently to translate this technology to the analysis of patient-derived T cells. So here we are looking at the morphological signature of T cells isolated from WAS patients and patients with mutation in ARPC1B and WDR1. And we are now not selecting a few features, but we are really extracting something like 500 features that stem from the cell profiler pipeline. And we set the values to 0 for healthy donor cells that we use as a reference. And what you see here in blue would be features for which values are downregulated in the WAS patient T cells, in the ARPC1B deficient T cells, and in red would be values that are upregulated in individual features. So very clearly, we obtain very clear morphological profiles for those different disease groups, with the most effective ones for the ARPC1B patient T cells, followed by the WAS patient, a much milder deviation from a healthy donor for the WDR1 patient. So this we could also compare to what was happening when cells were stimulated only with ICAM1 as a way to stimulate motility. And we obtained quite similar picture in terms of the defects in our 3 patient groups. Most patients affected being the ARPC1B defective patients, followed by the WAS patients, and a very distinct signature for WDR1, which values that seem to be kind of opposite to the ones of the 2 other patient groups. So out of those complex morphological signatures, we can also automatically extract discriminative features. So we found F-actin intensity on a per cell basis and the major axis lengths of the cells to be 2 such very discriminative features to compare our different patient groups. And when now plotted one against each other, those 2 features help us to understand really what's happening in those patient cells. So here, again, I'd like to call this to stress that this is across thousands of cells. So what we see is that in the Wiskott-Aldrich syndrome T cells, they have less actin and are shorter. This defect is much more pronounced in the ARPC1B deficient T cells. And those features are not much affected in the WDR1, at least when in the context of the immunological synapse. But when you look at the context of cell migration with ICAM1, you see that there is a tendency for those WDR1 T cells to have higher F-actin content and to elongate a bit more than normal cells. So as you see, this pipeline allows us to very much discriminate patient cell morphologies, to extract meaningful features to understand the biology of those individual pathologies and individual patients. So what's next? So we have recently assembled a library of expanded T cells from multiple patients. So we have about 30 patients with various mutations. And we are also comparing them to a large cohort of healthy donors. So we are in the process of performing the morphological profiling of the cells over different stimulatory surfaces. And in parallel, we have also developed high throughput functional assays, such as assays to measure the killing activity of our cells. So the way we are planning to assemble the data is by plotting the individual morphologies from patients and disease groups on a TSNE type of graph in a way to look at similarities and dissimilarities among different patients and disease groups. And then, in addition, we plan to overlay to that morphological profiling the results from the functional assays, such as the cytotoxic assay, in a way to relate defects in morphology to functional defects. So what is the outlook of this pipeline? We think that we will be able to compare disease severity, classify disease entities, and also we think that we will be able to prioritize variants from patients with undiagnosed disease. So this approach is also a very efficient way to identify disease-specific cellular readouts that we plan to use for drug testing. And of course, we think that we can extend this pipeline beyond T cells. So I'd like to thank my contributors who participated and did all this work. So in particular, Raisa for all the super resolution microscopy imaging, and Anton and Diola for the high content imaging. This work has been done in the environment of my lab in Toulouse, in particular with the help of Sophia Lahr, and in Vienna with the help of Kan Borsuk and all his team. And I would like also to thank all the specialists from the field of immunodeficiencies and clinical contributors. Thanks a lot for your attention.
So good morning, everybody, and thank you for this invitation of presented ARPC1B related diseases features. I have nothing to disclose, and I will start presenting the clinical characteristic of two patients that we follow during the year, and then I will go through what we collected as a clinical phenotypes and some novelties in basic studies on ARPC1B function. So we follow Giovanni for many years. Giovanni presented with macrophage syndrome in the first months of life, triggered by CMV infection. You see a rash that was linked to the presentation, and then he developed a chronic CMV infection, some other skin infection with pustulosis linked mostly to Staph aureus and stunted growth. On the immunological side, Giovanni presented a mild lymphopenia with an expansion of gamma delta lymphocytes that were oligoclonal. Response to mitogen was normal. There was a light defect in the anti-CD3 and CD28 proliferation. We thought because of a gamma delta expansion, CMV infection, and immunodeficiency had some leaky SCID mutation in SCID genes, but we didn't detect any, especially in REG1, 2, or other. Giovanni then later in life presented with some recurrent flares characterized by inflammation, fever, and enlarged lymph node or a pseudotumor. Here you see infiltration of the diaphragm muscle. These flares were very painful and it was in very bad clinical condition, overall condition. The other kid that we follow is another Giovanni. The other Giovanni presented with a different phenotype. He had a presentation with a bloody stool and inflammatory bowel disease-like, resistant to immunosuppressant, and diagnosis of protein milk intolerance. He presented with eczema and very severe lung infection. Here you see a pneumatococcus from Staph aureus, and later in life, recurrent warts that was diffused to hands and arms and thorax. He also presented thrombocytopenia, mild thrombocytopenia, increase in IgE and IgA, and a mild effect in IgG and IgM. He had also a mild lymphopenia, with CD8, CD4 lymphopenia, and expansion of double negative T cells. Giovanni was treated with some immunosuppressant, and we gave him IVIG and bacterial prophylaxis for the infections. We rolled out together with Silvia Giuliani in Brescia with Scott-Aldrin syndrome defect, obviously, both in the protein expression and gene sequence, and with defects, but we didn't find a mutation. So we submitted both patients for extended genetic screening. We sent the DNA of the more inflammatory one to Gigi Notarangelo in Boston at that time, and I forgot to tell that the parents of this patient were consanguineous, so it was highly likely that it was a recessive defect. And the other patient was analyzed by Alessandra Aiuti group in Milan, with a target panel of known and not yet described gene linked to immunodeficiency. At the time, then at ESID, when the result came back, there was a gene that was in common mutated with a homozygous mutation was ERBC1B, and then at ESID several other groups reported other patients. So we decided to combine all the patients reported so far with Taco Kuiper in Amsterdam, with a very nice collaboration. And in our report that was in 2018 in Jackie, we collected all the clinical and immunological features. And in the last years, 2-3 years, other groups presented other patients, other families. And here you see there are both consanguineous families and non-consanguineous families, and only one affected kid or more than one. So these are the mutations that I was able to retrieve on published papers so far. There are at least 24 published patients, and I received some other personal communication of at least 4 patients. And you see that the mutation are different kind of mutation: missense, splice size, nonsense, and frameshift, and are all over the gene. These are the exon and the introns of the gene, ERBC1B gene. Most of these mutation cause, or all of these mutation cause, a decrease in protein expression, the one described so far. Here you see a 3 patient. This is immunofluorescence with ERBC1B in red and affecting in green. And you see a decrease or a complete absence in some patient, and this is the control effect. This is another interesting feature on protein expression, that ERBC1B here is in green, ERBC1A, that is an isoform, is in red, and this is a control on a patient, a control on a patient. So on, whenever the ERBC1B is not expressed, as you see here, there's an increase in expression of ERBC1A. We don't know the meaning of this. Clearly, this is not compensated because these are patients, so with pathologic feature, but we reported this. So what is the clinical presentation of patient? I collected, so the first report that we did were about 14 patients. This is data from 24 that I got from the literature, and most of them present infection. All of them present infection. Most of them present lung infection. You see, this is 100% of the patient. Most of them present also growth failure. This is an interesting feature that I think deserves some more studies. We don't know if this is only related to infection, because several patients were tested for GH secretion in response to arginine or other stimuli, and several patients would search for skeletal dysplasia feature that were not found, but still I don't think it's just related to infection rate. Allergy with eczema and food allergy is present, and a combination probably of the light blood disorder that this patient have and autoimmune tend to present early in life with enterorrhagia. So also severe bloody stool in the first months of life. In terms of infection, this patient presents especially lung infection. Here you see a few patients that present with pneumatococcus, and also skin infection with abscesses, warts, mollusco contagiosum, and recurrent otitis media. There are at least 5 patients with a chronic or severe CMV, chronic CMV or CMV pneumonia, and a couple of patients that died because one patient for sure, and the second one probably because of adenoviral infection. In the pathogen, most pathogens are bacteria. 84% of patients present bacterial infection, but also virus are a problem in this patient, and they tend to have problems in controlling virus. This is data only a few patients because we didn't receive a complete description from all the other one, and from the literature it's not clear if they describe all the pathogens. So I'm just presenting what we know for sure that were characteristic of this patient. And you see Staph aureus and Pseudomonas and Mollusco contagiosum CMV might be a problem. There doesn't seem to be a major problem with the fungi. There is a problem for sure on the immune dysregulation, presenting with allergy and inflammation. Here you see a patient with a severe eczema that is present in most of the patient and is a clear characteristic, and food allergy and asthma I already told you about it. There are other features of immune dysregulation because it's both autoimmunity and also some inflammatory feature already described by our patient, but most of the patients present with several patients present with colitis and also skin vasculitis. And this is really characteristic. This is a biopsy from colon from a patient. This is very characteristic, the skin vasculitis, because in the early months of life tend to present in this way, so with this feature, and tend to also evolve in necrotic and ulceration, necrotic ulceration. And when biopsied, we also biopsied out the patient, and the presentation was always as a leukocytoclastic vasculitis with infiltration, you see a neutrophil vessel. And this lymph node enlargement have been reported in several patients. It doesn't seem to be similar to ALPS patient as a lymph node increase. Feature of hematological and immunological feature include thrombocytopenia, as already said, but also increased eosinophil and IgE, and there's an increase in IgA also with normal IgM and IgG. There's a decrease in CD8 lymphocyte, and we're going to go through this in a recent paper that clearly nicely described this some pathogenic feature of these cells. There's an increase in B lymphocytes, both in the percentage, as you can see here. This is effect showing CD19 and CD20 expression. You see in one patient an increase in percentages, but also in absolute numbers. And this has been reported in several patients. There's a defect, a mild defect in proliferation. As I told you, is particular with using anti-CD3 and probably with a low concentration of the anti-CD3. Here you see a patient on the right side of this table. This is a patient with increasing concentration of anti-CD3, and this is a control. You see the difference, and this difference is reversed when the patient was transfused with a lentivirus containing RC1B gene. This was performed by Immaculata Brigida in Alessandra Iuti's lab. We found a defect in NK cell degranulation, CD107 expression in 2 patients. It was reversed, partially reversed by IL-2, and this is similar to what has been shown in Wiskow syndrome, but probably needs to be confirmed in other patients if this is true or not. So I already told you what can be the mechanism, some of the mechanisms that are responsible for the infection of this, for recurrent infection of this patient, a severe infection. So there's a defect in CD3 mediated activation and T-cell development and differentiation. I'm going to show you. There's a defect linked, a typical defect linked to actin dysregulation. There are cell migration, immune synapse formation, and there's a specific defect in CD8 maintenance cytolytic activity shown by one group recently. So Somek et al. In JI in 2017 nicely show how RC1B is required for proper T lymphocyte development. This is a zebrafish model. This is the eye, and this is the swim bladder of the fish. Here you see LCK staining or GFP staining that is localized in the thymus. This is from above. Fish has two thymuses, and here you see when you with RNA interference, you decrease the expression of RC1b or complete abrogate the expression of RC1b, you don't have any more signaling in the thymus, so the thymus are not formed. This is the same experiment, but with immune in situ hybridization, and again, staining for LCK RNA. And here you see that the morpholine has abrogated the expression of LCK in the thymus, and with a co-injection of RC1b human RNA, you have a partial, at least a partial reverse of the phenotype. In terms of differentiation, I already presented the spectrotyping data on gamma delta cell. This was confirmed by Brigida and by Somek in their publication. And this is especially true for TCR beta and TCR gamma with expansion of them, some clones. This represent, each area represents a single TCR beta, and the size of the area represents the expansion of the clone. You see an healthy control with a several, with a complete repertoire, and here you see a difference in effect. And this doesn't seem to be the case for B cells. And again, the importance of R2B expression in lymphocyte is underlined also by a phenomenon that's typical in Wiskott-Aldrich syndrome. There is a revertant, the presence of revertant, so where there's a second mutation that revert the expression of the protein. And here you see in CD8 cell of our patient where there is an expression of RC1b in part of the cells, peripheral blood cells of CD8 cells, and especially is in terminally differentiated T cells. And this reverts probably partially the phenotype in these cells. We don't know if they're functional or not. What is the effect of an altered actin cytoskeleton? This has been proven in different other diseases related to actin, but also in this patient, there's a defect in lamellipodia formation, so some protrusion, small protrusion. They tend to form bigger, longer protrusions that are called filipodia that you can see here, and this was also confirmed by Ranzavol et al. In JCI lately. And this is a beautiful movie from JCI publication where you see healthy donor on the left and RC1b patient cells on the right. These are CTL cells, and you see there's a reduced motility and the formation of this filipodia that doesn't happen in a healthy donor cell. This has been quantified, this defect in migration, by several groups with several stimuli. And here you see a complete lack with SDF1, for example, stimulation in % of migrating cells. Immune synapse formation is impaired. And here on the left, you have a healthy donor cells, T lymphocyte, and on the right column, cells from a patient. In red is RC1b expression, and in green is F-actin. This is a target cell, so P815 cell. And you see that when these target cells are coated with anti-CD3 beads, as in the lower panel, there's a clear interaction between T cells and the polarization of both RC1b, the localizing the immune synapse, and of F-actin. This is already known, obviously, but RC1b tend to polarize and localize into the immune synapse. And immune synapses are not really formed in this cell. And again, there's a beautiful movie showing that in this paper from Zavala et al., where you see an healthy donor cell, CTL, making a proper immune synapse with target cells and RC1b cells forming this long filopodia and not being able of forming an immune synapse with target cells. So in their publication, they studied CTL function. They showed that there's a defect in the lytic function of these cells. Here you see RC1b deficient cells that are unable or defective in lyse target cells where healthy donor behave normally. And very interesting finding that they also present is that when you isolate with beads with a negative selection CD8, so you select CD8 without touching the CD8 and stimulate them for 13 days, healthy donor cells keep their phenotype with CD8 expression, and they don't express CD4, obviously. But RC1B patients tend to lose the expression of CD8. They don't express CD4. They are not TCR gamma delta. They clearly show in the publication. So they lose the expression of CD8. And CD8 is not completely lacking, but is intracellularly located. So at least in a percentage of 10% of these cells that lose the CD8 expression, CD8 is located intracellularly. Here you see the patient, and here you see a healthy donor. And so CD8 is present, but is intracellular. As well know, membrane protein like TCR and CD8 and CD4 have to relocate from the membrane and intracellularly, and then translocate into the membrane. Probably they show that this pathway is abrogated in patient cells, and there's a staining, intracellular staining of both CD8, so there is not recirculating in the lower panel patient cells, but also of other proteins that are known to recirculate in this way, such as GLUT1, that is a glucose channel, and here you see a punctate staining inside the cells. So they clear there's a problem in circulation, and they speculate that there might be a link between this glucose channel mislocalization and the survival defect that they see in these cells, and also in proliferation, since cell needs glucose and energy to proliferate. Here is a healthy donor CD8 with a proliferation dye that is diluting the dye going from right to left, and this is the unstimulated cell. Here you see in patient, almost not have any proliferation. Again, what we know about autoimmunity and inflammatory mechanism. We know something about autoimmunity, so we know that there is a defective Treg function. We presented in our report a couple of years ago, and this is work done by Eloy Quadrado in Taco Keeper lab. There's a defect probably in expression of marker of the regulatory T cell, and there's a clear defect in function. Here you see responder cell unstimulated and stimulated, that they proliferate, so they dilute the dye from right to left. And whenever you add cold culture, the responder cell with the Tregs in different concentration, you have a suppression of this proliferation. Here you don't have this in patient cells, and this has been done by with several different patients, and it was very consistent. So there's a defect in Treg function. This was already shown in Wiskott-Aldrich patient, and is confirmed also in this patient. We don't know much about inflammation. Serum cytokine, we measured them in our patient. During acute phases, we found an increase in 6CL10 or IP10. We don't know if this is linked to type 1 interferon, type 2 interferon activation. We didn't get consistent data of type 1 interferon activation in the peripheral blood of these cells, of this patient. There's a slight increase in IL-6. As I told you, this patient tend to have some blood disorder, aggregation probably, because of a defect in platelets. So there is a defect in number of platelets and defect in function. Platelets tend to be smaller in size, at least the diameter. In this patient, these are RC1B patient. This is both patient, as published by Carr et al. And confirmed by other group, also by Alessandra Iuti's group and Anna group. And the platelet also tend to lack some protein that they normally express. So this is a normal expression of tubulin, P-selectin, TSP, and here you see some cells, some platelet lacking the normal expression of this protein. And they also do not express properly granules. They have defect in granule content. These are 2 patients and a control with Wiskott-Aldrich patient control. And they have defect in activation upon stimulation with ADP or thrombin in terms of expression of PAC1, as shown here by Anna Villa's group. There is some novel data regarding something that is yet unpublished that yesterday Caterina Cancrini presented, and I'm just gonna we did a nice collaboration on that with her group and Dottoressa Chiriaco and Di Matteo in Bambino Gesù Hospital in Rome. So it was very recently shown that ARP complex and viscoprotein syndrome protein and nuclear actin are required for double-strand break repair, at least in the homology-directed repair. And here you see a gamma-H2AX staining upon irradiation of cells. You see a colocalization with WASP. They presented very nicely that also they colocalize with ARPC1B, but they are too complex, but they do so. This is a chromatin immunoprecipitation experiment where you see that there is a WASP location bound to the DNA upon irradiation in different phases of the cell cycle, and ARP complex only bind in the G2 phase. This is upon irradiation of cells. Before the diagnosis, they have a patient with ARPC1B deficiency syndrome that they follow several years. And before the diagnosis of ARPC1B deficiency, it was tested for DNA damage susceptibility, and here you see in fibroblast that there's an increase in gamma-H2AX foci upon irradiation in this patient compared to control, and this was true also in LCR-EBV LCLs. This is their patient, this is our patient, and this is especially true in G2 phase. So I'll tend to recapitulate what this group described in animal models and cell lines, and these are the foci staining, gamma-H2AX staining, and this is the quantification that clearly show this increase. So what is the outcome and the therapy to conclude it? It is not clear yet, so we need really several data probably. I try to put together what has been published and what I heard from personal messages. So life expectancy is impaired, but it's too early to say how and how much. There are, out of 33 patients, there are 7 that are deceased, 6 very early in life before age 5, some in the first months of life, 1 a 24-year-old for pneumonia. Median age of patient alive is 12-year-old. Obviously, this is the age of reported patients, so we don't know if they're still alive and how are they doing. IVIG and Bactrim are important for infection prophylaxis. They tend to require steroids or immunosuppressant for autoimmunity and inflammation. Several patients have been transplanted, but we don't know the outcome of this. Please, if you are willing to collaborate, our BMT group, Stefano Giardino, is trying to collect data on that. Our experience is on our 2 patients that have been transplanted with haploidentical alpha beta CD19 depleted cells. Both patients, this is the condition in both patients, recovered. One patient 6 years after transplant is doing well. The other patient, the second patient, 1 year after the transplant, developed a JC-related PML that lead to complete neurological loss of all function and coma. We treated the patient with a JC-specific CTL that resolved the coma. The patient required most of his function, still have some extrapyramidal problem. And lastly, I have to thank all the patient and the family that collaborated to these studies, my group, Marco Gattorno, Roberta Caorsi, and the group that collaborated provided data from Amsterdam, from Milan, Alessandra Aiuti, and Immacolata Brigida, Anna Villa, and Silvia Gigliani in Brescia, and all the other groups that work in this disease. Thank you very much.